Hi, everyone, and welcome to episode 602 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and this week we are excited to be rebroadcasting our interview with the wonderful Dr. Rachel Wong, originally released on November 12th of 2014. For those of you who don't know Rachel, she is an inspirational scientist who has developed and applied a variety of microscopy techniques to investigate the connectivity of neurons in the retina. Her lab has been working on understanding how retinal cells may reform functioning circuitry after damage. In this interview, Rachel talks more about her research, her life outside the lab, and some of her formative career experiences. So listeners, I hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am delighted to be here today with Dr. Rachel Wong. So great to chat with you today, Rachel. How's everything going?
Very well, thank you. It's summer in Seattle, so the sun is shining and we're all feeling good.
Wonderful. Well, great to hear. And I'm excited to introduce you to our listeners today. So listeners, Rachel is a professor in the Department of Biological Structure at the University of Washington. She received her PhD in Vision Neuroscience from Australia National University. And afterward, she served as a research associate at the National Vision Research Institute of Australia. Rachel then conducted postdoctoral research as a CJ Martin Fellow at Stanford University and then an RD Wright Fellow at the Vision Touch and Hearing Research Center. She then served on the faculty at Washington University in St. Louis before joining the faculty there at the University of Washington. Now, Rachel is here with us today to tell us all about her journey through life and science. So today we are excited to get to know you, Rachel, both as a scientist, but also as a person. So can you take a few minutes here to tell us what you like to do when you're not doing science?
Well, I think my other passion is music. And right now I'm trying to learn the violin. It's challenging for someone who's much older, but that's where I'm spending most of my time right now. And the other thing is to look after members of the lab because that's really my family.
Oh, wonderful. Well, Rachel, we're going to have to play some duets sometime. I'm a violinist myself.
Oh, excellent. Well, I'm only in book 2 of Suzuki. It might take a while now.
We can start with some easy songs. I like that. I'll probably need a refresher myself. Very good. Well, great to hear that you're exploring a new instrument in music. The violin's a fantastic choice. And great to hear that you've got this family environment in your lab and you help take care of all those students under your wing. So, Rachel, great to hear about how you like to spend your time when you're not doing science. But now I'm going to ask you to tell me a little bit more about that scientific side. So can you tell us how you describe what you do to someone who's not in science?
I think our biggest interest is to try and understand how neural circuits in the retina, and that's the light-sensitive part of our eye, we really like to know how these circuits are assembled properly during development. And also more recently, we've got interested in trying to understand how these circuits can become rewired or repaired in disease. The latter part, of course, is extremely challenging.
Very interesting. So what sort of disease states are you looking at?
I would say in any retinal diseases where you have loss of cells like photoreceptors or the retinal ganglion cells that really are the major cell types that send information to the brain. So if any of the input or output cells of the retina die, then essentially you're blind.
Interesting. Well, it sounds like you are answering some fascinating questions there in the laboratory, as well as juggling some amazing hobbies outside the lab. And I'd like to talk to scientists about what motivates them. What are these driving forces that help compel them to get everything done in just 24 hours in a day? So can you tell us, Rachel, about a motivational quote or maybe a saying that you love to keep in your mind to keep yourself focused and motivated?
I think this is a quote that my father passed on to me when I was in primary school, and it has stuck since. And it's procrastination is the thief of time.
Really? I like that.
I think ever since I was 7. In other words, you better do your homework now because there'll be more tomorrow.
Right. Well, that is a good lesson to instill early on. Fantastic to hear about this quote. So is this something that you tell your students now?
Yes, I'm not so sure how effective, but I am trying.
Well, that is a great motivational quote. Certainly important for life in general, but definitely with these applications for science. You're exactly right. If you don't do your homework, if you don't write your paper or do that experiment today, it's going to be there tomorrow doubled, I think. So great to hear about this motivational quote that you have. And I'm curious about some of these people who may have motivated you or inspired you over the course of your life and your career. Can you tell us about some of these role models you've had?
So I'm glad you mentioned role models, because certainly I think one's journey in anything is shaped by many people for different reasons. So I would like to name a few, although I've worked with many, many wonderful people over the years. And I think the first person who was really extremely influential in me going on for a research career, taking on this research path, is a guy called Herbert Bolton. And he was actually professor of physics at Monash University, where I did my undergraduate degree. And I think what Bolton really taught me was not to be afraid to ask questions. This was in a physics class where I was the only female at that time. And this was important because one has to ask questions as really a first step towards trying to make a new discovery, whether it's a personal discovery or whether it's a broader scientific discovery. So Bert Bolton was very important, I think, in helping set me into the research path. And then in my graduate days, I gained really the passion for retinal research from my advisor, Abby Hughes, who also helped me to develop critical thinking skills, which I think we all need when you are in research. And finally, I'll say during my postdoc years, my advisor, Carlos Schatz, who has contributed significantly to understanding of brain development, really was the one who introduced me to the world of neuroscience. And I learned from her how to recognize the broader implications or significance of my findings, and also importantly, how to communicate my work to the general public. So, each one of these advisors, I would say, imparted in me something different, but I think all of these factors were important in trying to shape, oneself as being a hopefully productive investigator.
Certainly. Well, Rachel, it is great to hear about these qualities, these traits that your mentors instilled in you. I think starting at the beginning, not being afraid to ask questions is so important in science. And oftentimes you feel a little silly because there's some questions where you think, I should know the answer, but I don't. And you have to ask those questions. It's very important. I love when you're kind of moving through these different mentors that you've had over the years, learning critical thinking and how to communicate your science and the importance and significance of your work. I think these are all huge lessons that scientists need to learn that you can't really pick up from a textbook. So it's great to have those role models there to help you. So wonderful to hear about these people along your journey. Now, can you take us back next to the very beginning, Rachel, and tell us where your interest in science began? Maybe one of your earliest memories about being excited about science.
Well, it was definitely a little later than 7 years old. Studying the piano then, and there were too many things to do for piano that I wasn't paying attention at school.
Right, you had to focus, yeah.
But I think I've always been curious about how things work in general. So even in secondary school in Malaysia, I was born in Malaysia, I was always bringing in samples to view under the microscope during lunch breaks. And this was actually very much encouraged by my science teacher at that time. And I remember that I would go to the creek next door to the house, although it was really a large drain. Mm-hmm. And I would pull from it all sorts of things. I'll take it into the lab and try and figure out what they were and what they did. So this was not so encouraged by my mother, of course.
Right.
Down to the so-called creek. But I think it taught me a lot about microscopy. And I, at that stage, was not thinking about becoming a microscopist, but more thinking about medicine. So in high school, I became interested in medicine, perhaps pursuing a career in, of all things, ophthalmology. But I also became very interested in physics. And when I went to Australia for undergrad studies and majored in physics, I found myself being more and more, lured by research, and not so much on questions that concern physics, but on biological questions. And so in my honors year, I was very fortunate that my physics advisor allowed me to formulate my own project and come up with something that, I could propose that seemed like a reasonable question using physics techniques but answering a biological question. And nowadays, of course, we would call this interdisciplinary science.
Right.
But neuroscience didn't exist as a discipline at that stage. So I was very lucky that he allowed me to do this and for me to go around the entire university to ask a biologist, at that time a biochemist, if I could please work with them. So I started working on lipid bilayers, particularly blood cell membranes on patients that had muscular dystrophy, and I could apply X-ray and neutron diffraction techniques on this. Spent a year in a nuclear reactor, actually, in Australia.
Oh, wow.
Discovered that I loved research, but I didn't want to spend another 4 years in a nuclear reactor. So I moved on to do a PhD, as you mentioned, at the Australian National University, where, again, I didn't really get onto this path that I am currently so quickly. I really started my PhD in the math department, but I wasn't so much interested in equations, let's just say, and I wanted to do experiments. And so eventually I was introduced to a wonderful anatomist in the medical school, and I started to work on the retina, and the rest is history. I have been working on the retina for a long time. And I think sometimes when I'm introduced before talks, people are surprised I'm still working on the retina.
There's still more to search for.
And so since then, I got my PhD from ANU, was very fortunate to be sponsored by the Australian government to work at Stanford with Carla Schatz. And then went back to Australia for a short stint and got my position at WashU in St. Louis, where I think that was a tremendous grounding place for me, where I really learned, being amongst just some incredibly supportive senior colleagues, just how to do science. And from then on, I was there for about 12 years and moved to my current position in Seattle at the University of Washington. Still confuses people.
Right.
Things have also been really good, and I have wonderful colleagues here also. So that's my kind of short journey, but it's over 20 years now, so maybe it's not that short.
Well, Rachel, it sounds like you have had a wonderful journey through the world of science. And I love always hearing about how your interest in science began. I like that you're describing, this general curiosity about how things work, and then getting your hands dirty in the creek, pulling samples out and looking at them under a microscope. I love that. That is wonderful. And certainly blazing trails in terms of this interdisciplinary research you were able to do, bringing physics and biology together. That is fascinating. So can you tell us a little bit more about what it was like working in a nuclear reactor?
It's very quiet.
Really? That's surprising to me.
Yes, it was extremely quiet. High security, very quiet. The food was terrible.
Really? Was there just a cafeteria?
Yes. But it was an opportunity to try and apply technology to the approach had never been done. So I learned from the very beginning, I guess, to ask the question first and then think about what approaches might be best used to try and answer the question. So that was good training.
Certainly. Well, we have hit some of these amazing high points along your journey as a scientist. Rachel, I feel like we've hit all the wonderful aspects of your career. We haven't yet touched on some of the struggles, some of the challenges, but I know every scientist goes through periods where, it just seems like everything's going wrong, where you're either really struggling with something or maybe have a major failure. Can you take us back to one of these times and tell us what happened and what are some of these steps that you took to really get yourself through that tough time?
Okay, so I think when experiments don't work, there's always a reason. If technically, for example, you've done the experiment so correctly, but the answer isn't what you might have expected or hoped, but that's all part of discovery. So those times when things don't work, they don't really bother me because I think they make you learn something.
It's part of the process, right?
In fact, it's giving you an answer, and you have to pay attention to that. I think for most investigators, as they go through their career, they are always going to hit some so-called failure, if you want to call it that. And I would say I've been generally quite lucky. This is with regards to funding, because that's a major factor when you do research nowadays. Mm-hmm. I think perhaps one of the most stressful times was not getting the renewal of my first NIH grant and wondering if, one can go on with this. But I was at WashU at that time, and I had some incredibly supportive senior colleagues in the chair of department that really assured me that this is quite normal. And if you didn't go through this, something was actually not quite right.
Right. You were too lucky.
It was really a setback, but a way to reassess your thinking and your plans. And so I learned from that how to focus on moving forward rather than sitting on and feeling bad about what might potentially be a setback, but trying to learn from it. And I think ever since then, even though every grant renewal is traumatic in its own right, one learns that you can only give it your best and certainly do your best and hope for the best. And it hasn't been too bad.
Well, thank you so much for sharing these experiences with us today, Rachel. I feel like your optimism is contagious. I'm feeling better about things myself here. Well, and I think that is such a great approach to take to these situations, is to try your best, go out there, know that failures happen. And if it happens to you, it's wonderful that you've had that network of colleagues to be able to support you, to be able to help you out. So fantastic to hear about how you get through the tough times in the world of science. So we don't just want to talk about the difficult points in your career, though. Can you tell us next, Rachel, about one of these success stories? I love celebrating the successes in science because I think, with all these challenges and struggles that you have, it's really important to celebrate those successes, whether they're the big ones like grants or papers or even some of these smaller ones that come along the way. So can you share one with us today?
Okay, I think that there are maybe two aspects, and if I can touch on them both.
Sure.
One is, I think, every time a project comes to fruition and you write up a paper, I think that's a success. And I don't think I can put any one particular finding well above any other than, findings that might spin off and produce more ideas and perhaps open even a small field. I would say the other success, and maybe more important than each of the discoveries, is being able to generate and support the next generation of young scientists. And I think if one can help, seed the approaches or way of thinking to help the next generation succeed, I think they might discover something far greater than one might be able to do, right?
Certainly.
That's actually important, and I'm very proud to have had many people in my lab doing well now. And hopefully that will be encouragement to members and also other scientists out there.
Absolutely. Well, I love that you have these, more traditional successes, making the discoveries, getting the papers out there, but also these mentoring successes, enjoying the process of training the next generation of scientists and really being a great mentor to them. So, Rachel, was this something that you kind of always have known you wanted to be in this mentorship role, or was this something that you've enjoyed taking on that responsibility as you've gone through your career?
I think I've always enjoyed working with people, and although, focusing on the science, planning the experiments, knowing what your goals are all extremely important. But I think I've always enjoyed working with the people who are actually doing the work in that sense.
Wonderful. Well, can you tell us next now? We talked about, the process, your journey through science. Can you talk about in a little bit more detail one of these projects that you're working on now in the lab that you are so excited about?
So one of the projects I think that we are spending quite a bit of time thinking about is how do circuits that get disrupted in disease become rewired or reorganized or reestablished if you could put the cells back. So we've been using zebrafish as a model system in addition to our mammalian model systems to try and understand this process. In other words, whether there are limitations in trying to rewire circuits that are disrupted. And I think we are learning a lot concerning the regeneration process itself. And one of the major projects I think we've undertaken allows us to use live cell imaging to watch in the eye of living animals as the regeneration occurs. So we can actually watch cells be born again, so to speak, and rewire with the existing neurons in the retina. And I think this will trigger a lot of interest and also help us understand perhaps the cellular processes and the steps that take place that will eventually allow us to design strategies to help us wire up the mammalian visual system, which is not so fortunate as in zebrafish, where we don't generate our own cells, neurons, once they die. But I think we can learn from other animals that do. how to do this job.
Well, that sounds like an incredible project. So when we're talking about these cells being reborn and starting to rewire, what sort of time span are we talking?
So fortunately in the fish, this occurs in days.
Wow. That is phenomenal. So you're able to check back at different time points throughout this, couple day span.
Yes. You can label the cells in transgenic animals with fluorescent proteins, jellyfish fluorescent proteins, and their variants with different colors. So it's like a coloring book. And you can label each type of cell and their connections as well with different colors. And you can watch this sort of choreograph, move together and how they might rewire. You can follow exactly the same cells after they've been made and watch how they tap back into the network.
That is phenomenal. Is this something you ever perhaps imagine you'd be doing those early days when you were pulling samples out of the creek and looking at them under a microscope?
No, there wasn't much color then either.
Right. Well, Rachel, this is an amazing project and really fascinating to even imagine being able to watch these cells sort of being reborn, rewiring, reconnecting. So it's exciting to hear about some of these projects that you're working on that you were thrilled to be a part of. And I know scientists stay so busy with all the research that they're doing in the lab, as well as juggling the things that they like to do outside of the lab. It doesn't always leave a ton of free time to kind of sit back and relax, maybe pick up a good book. But I know a lot of scientists are avid readers, and I've gotten some incredible book recommendations from people we've had on our show. So can you tell us today, Rachel, about a book that you've read and really enjoyed that you would recommend to our listeners?
So this might not be too surprising, but
I'll act surprised. No.
There's actually a series of books called Number One Ladies Detective Agency. Alexander McCall Smith. It's staged in Botswana. It's fictional, but it's a marvelous series because it's about solving a mystery. This is a private investigator, and she runs a little business trying to solve a local mystery. But what I find really good about these books is that not only is it solving a mystery, but at the same time, you learn a lot about life's lessons. And I think, each time I pick up one of these books, I would find that at the end of reading the book, I always find myself smiling. So it's both encouraging, heartwarming, as well as intriguing.
Well, that sounds like a wonderful recommendation, Rachel. We'll have to put those out on our website for our listeners to check out. And I love reading these sort of mystery, kind of suspense, heartwarming, though, kinds of books. I hate reading sad books, so I'll have to check this one out myself. So great recommendation. And in addition to some of the amazing work scientists get to do and some of the fantastic books you're maybe able to enjoy in your little bits of free time here and there, I know a lot of scientists get really excited about the opportunities that they have as part of their career to travel. And these travel opportunities may come for conferences, to work with collaborators, things like that. Can you think back now, Rachel, on all of these different places that you've traveled for science? And can you tell us about your favorite place? And what was it about this place that really made it so memorable?
So I think in all my travels, each place scientifically, of course, taught me something new, and meeting new colleagues, that's always wonderful. And so one makes a lot of friends and also learn a lot in terms of what they're doing scientifically. But I think my most recent trip to Israel was the one that was really memorable. And I think the reason for that is not only the science, of course, at the meeting, but that I realized that the discoveries we made are really still pretty small in the history of mankind. I think visiting somewhere like Jerusalem really reminds you that you're part of this thousands of years of civilization, and there really is a lot to do. And I came away, I think, saying that I'll never forget that trip.
Well, that sounds like an incredible experience. I know, spending most of my time in the United States, things are so young here. So when you go to Europe, when you go to the Middle East, it's just incredible to be surrounded by that culture and just the amazing richness of the societies that lived there before. I think that is fantastic. So how long were you in Israel?
It was only about a week, actually.
Interesting. So was there a particular site that you saw that really struck you while you were there?
I think it really was Jerusalem, because the layers of civilization and, you get a sense of the present with the past and the hope that one learns from the past what to do in the present. I think that really kind of hit home.
That sounds like an incredible travel opportunity, and certainly one of the places I've never been, so it's on my list of places to go. Well, it's great to hear about these places that you've been able to see. And you hinted a little bit at how wonderful it is to, make colleagues out of a lot of the people that you meet at these conferences, and certainly the importance of the people in your lab and throughout these different research groups you've worked with over the years. And I know, there are some stereotypes out there about scientists, but I think a lot of the scientists that I've met and worked with are just amazing, creative, and oftentimes quite funny and fun people. So the research is very important. You take it seriously, but you like to have fun too. And I think working with so many brilliant people, you get some quirky traditions that spring up, or even just funny memories that you can look back and laugh about. So can you tell us today, Rachel, about one of these traditions or memories that you experienced?
Well, I guess no one in my lab would want me to call them quirky. And there are none. They are all very ordinary people, but also amazing in their own rights. I would say maybe one common thing amongst everyone that's ever passed through my lab is that we all like to eat. Mm-hmm. And so food is always a big topic of discussion in the lab and in any of the lab planning other than the science.
So do you guys go to lab lunches, or what are the occasions that warrant food?
We bring in food and share, and I often go to dinner with the lab, or we can cook together.
So do you have a special dish, Rachel?
Nothing particular, but I think one thing that I will always remember is one of my postdocs showing us how to make udon from scratch, and it involved stepping on a bag of dough.
Oh, wow.
Kneading it that way. And so that was quite interesting.
So you had to eat something that you had walked on.
Yes, walk on the food first.
Yeah. Did it come out good, though?
It was excellent, yes.
Oh, wonderful. Well, I've never tried to make it from scratch, so I'm sure it's quite a process. That's incredible. Well, it sounds like a wonderful tradition to have, to bring in food to share with everyone, especially have international students or people coming from different cultures who have their own favorite dishes that they can share with people who may have never had it. So great to hear about this tradition. And I think food is really a common theme that we're seeing across a lot of different laboratories, whether it's celebratory lunches or people bringing in snacks, things like that. So great to hear that you're keeping that tradition going strong.
Yeah, type of science is also about cooking, so it matches well with scientists, I guess.
Absolutely. It's the chemistry aspect of it. You got to do kitchen chemistry. I like that. Well, Rachel, that is fantastic to hear about how you and your lab mates bond over food and cooking and different opportunities for sharing food. So I think in addition to talking to scientists about your research and all these questions that you're able to work on and that you've answered over the years, I know scientists, we often get a kick about talking about the what ifs, the hypotheticals. So can you think today, Rachel, about if we took away all the restrictions, if things like funding and staff and technology and feasibility were not an issue for you, what is one research question that you are just dying to know the answer to?
I think right now we would really like to understand how to rewire a damaged nervous system, not only, to try and help people that have trauma, but I think being a developmental neurobiologist, I'm curious how a mature nervous system can deal with reorganizing its circuitry when all the developing cues may be gone. So I think focusing on trying to understand rewiring in the adult nervous system is still a big question for us.
Certainly. So if you had to pick one of these limiting factors, things like funding, staff, technology, whatever, is there one that really stands out in your mind as something that's keeping you from getting to that answer?
I think it's technology right now. I think the funding is quite good in this area of regeneration and rewiring of circuitry, but we still need better, molecular cell biological techniques to understand how cells do their day-to-day job.
Certainly. Well, I think that is an amazing research question to want to know the answer to, with definite applications to health and in medicine, things like that, people who have damaged nervous systems. So great question. So hopefully the technology will continue to progress towards getting us there. Well, Rachel, I love thinking about these big questions with scientists, and I think it's fun to chat with them, with your colleagues, with your mentors over the years. And in addition to talking about research, I think a lot of these people you work with and spend time with over the years impart some really great advice. So can you share with us today one of these pearls of wisdom, one of the best pieces of advice you think you've ever received?
So I think this stems back to my undergraduate piece, which is I think we should not be limited by our current knowledge or expertise. If we feel that we do not know how to do something technique-wise, we wouldn't be able to answer a particular question. I think the key is to figure out then how to do it and then go for it.
That is fantastic advice with, great applications to science. I mean, you have to figure out what to do, but I think the action component is huge. You can't keep putting it off. You have to take action. You have to address it.
Right. I think you also don't want to be afraid to take on an approach where you may not be the world's expert or even have tried it yourself. But if you know what needs to be done and how to control for it, then you should go for it.
Be brave in your science. Well, Rachel, is there any other last piece of advice or maybe a last piece of inspiration that you would like to leave our listeners with today?
Well, let's see. I think I would like to encourage people to pick a research career. I think we should think about research not only about making discoveries or perhaps finding cures or helping to make life better for us who may have a disease. But I think in being a scientist, you also learn how to improve your ability to face challenges, whatever they might be. And I think also importantly, it also helps you become more aware of the needs of others. So I picked this direction, mainly because, as I say, I was curious about how things work. But I also found it challenging and a way to sort of improve who I am in general.
Certainly. Well, I think that is a fantastic piece of advice or something for people to think about going into careers in science. The discoveries are so important. Making progress in science is so important, but the process is so important. A lot of scientists are very results driven, thinking about the end goal, what they're working towards. But I think that process of self-growth, learning things about yourself is so important as well. Now, we've had some wonderful conversations today, Rachel. Can you tell our listeners how they might be able to reach out to learn more about you or your research?
So we have a website, if you just search for Wong Lab Biostructure, that will describe our current research and what we're about. I can also be contacted by email.
Fantastic. Well, Rachel, thank you so much for joining us today and sharing a piece of your story.
My pleasure. Thank you.
Certainly. It was wonderful to chat with you. And thank you to our listeners for tuning in today. If you love meeting amazing scientists, subscribe to our podcast on iTunes to make sure you don't miss hearing from your colleagues or your science heroes. Join us again next time for another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.